So there's really three main types of experimental designs. And so the first that we're going to talk about is called a completely randomized design. And then second, we're going to talk about something called a randomized block design, which is very similar to the completely randomized design. And then we're going to finish up with something called a matched pairs design. So a completely randomized design is fairly straightforward. Basically, it is an experimental design where each experimental unit is randomly assigned to a treatment. So like my migraine example that I was using was really a completely randomized design. I was going to randomly select which individuals get the placebo, which individuals get 100 milligrams, which individuals get 200 milligrams, and which individuals get 400 milligrams. So by randomly assigning who goes in each of those treatment groups, I am using a completely randomized design. Another example, a farmer wishes to determine the optimal level of a new fertilizer on his soybean crop. Design an experiment that will assist him. Okay, so step one, what's the purpose? The farmer wants to identify the optimal level of fertilizer for growing soybeans. So what does optimal mean? Optimal means it's the amount of fertilizer that results in the maximum yield, the most number of soybeans. So the response variable will be the yield, the amount of soybeans that are produced. Step two, what are the factors? Well, some factors that's going to affect crop yield will be, first of all, the amount of fertilizer. That's a factor that could affect the crop yield. Other things would be things like the amount of rainfall, so precipitation, the amount of sunlight, the way that we plant the soybeans, so how do we till the soil and plant the soybeans? What's the type of soil, like is it clay or what? The kind of plant we're using, okay, so what's the variety of soybean, the temperature, and so on, and so on, and so on, okay? So there's lots of different factors that we might have to take into consideration. So how many experimental units are we going to have? So we have to decide that for our research. So in this case, in this experiment, we're going to plant 60 soybean plants. So those plants are going to be our experimental units. Now that's a relatively small number of individuals, especially since we're talking about soybean plants. So list the factors and their levels. Well, fertilizer. This factor is going to be controlled, and it's going to be controlled at 3 levels. We wish to measure the effect of varying the level of this variable on the response variable, the yield. We will set the treatment, the level of fertilizer, as follows. So again, we're going to vary the amount of fertilizer on different segments of our sample. So in this case, we're going to have 3 treatments, so to speak. So treatment A, we're going to have 20 soybean plants that receive no fertilizer. So that's going to be the control group. Treatment B, 20 soybean plants are going to receive 2 teaspoons of fertilizer every 2 weeks. And treatment C, 20 soybean plants are going to receive 4 teaspoons of the fertilizer every 2 weeks. So that's the one we're really kind of controlling. Precipitation. Now, we can't really control the amount of rainfall that's going to occur unless we were to put these soybean plants like in a greenhouse. If we were going to put them in a greenhouse to grow them, then we could control the amount of water that they receive. Now, if we're doing artificial watering, we can control that even if we're out in a field. So we make sure that all of the plants are receiving the same amount of water if we're doing artificial watering. But in terms of like actual rainfall, we can't really control that. So what we can do though is plant all 60 of these soybean plants in like the same field so that whatever rainfall one soybean plant gets, the other soybean plants are going to get the same amount, okay, whether it's a lot or a little or what have you. The same idea with sunlight. We can't really control the amount of sunlight, again, unless we're in some kind of a controlled environment, like inside of a building where we're using false lighting, then we could control that. But assuming we're out in a field, we can't really control the amount of sunlight. But what we can do is plant these 60 plants in the same basic vicinity so that they are all receiving the same amount of sunlight. The method of tilling. We can control this. So again, we can till the soil the same way for all 60 plants, whatever the method's going to be. So we can control this factor by using Roundup Ready method of tilling for each plant. The type of soil. Again, we can control that by planting all of the plants in the same type of soil, which would be in the same kind of field. So again, certain aspects of the soil, such as level of acidity, can be controlled. In addition, each plant will be planted within a 1 acre area, so it's reasonable to assume that the soil conditions for each plant are equivalent. So we're not going to be planting one set of plants in one soil type and another set of plants in a different soil type. We're going to have them in the same kind of soil, in the same field. The type of plant. Well, there are many variations in soybeans, right? So what we're going to do is we're going to control that. We're going to use the same variety of plant. And just like people, even individuals in the same family have some variations, right? They don't all turn out exactly the same. Well, there's variations between individual plants that could occur. So to control for that, we're going to use randomization. We'll randomly assign which plant gets which treatment. And then last but not least, temperature. Again, this is uncontrollable, assuming we're out in a field. We could control it if we're in a greenhouse, but if we're out in a field, that's uncontrollable, but it's going to be the same for each plant as long as we plant them in the same vicinity. Again, that's an important step. Step 4 is really important, that we've taken into consideration all of the factors that can influence the value of the response variable. In this case, the yield of the plant. Step 5, randomly assign each plant to a treatment group, till the soil, plant the soybean plants, and fertilize according to the schedule that we laid out. So again, 20 plants get no fertilizer, 20 plants get 2 teaspoons every 2 weeks, 20 plants get 4 teaspoons every 2 weeks. We're going to go through our process that we set up. And then B, at the end of the growing season, we're going to determine the crop yield of each plant. So all 60 plants, we're going to meticulously go through and determine how many soybeans we actually produced. And then step 6, we would perform inference, which we'll talk about later on. We're going to compare the results of the 3 treatments, placebo, 2 teaspoon, 4 teaspoon, and then we're going to try to generalize that to the population of soybean plants if possible. Now, this figure that we have here is a pretty common thing to include. This is a diagram of our experimental design. So this is how it works. We're going to start by randomly assigning plants to treatments. We have 3 treatment groups, and so we're describing those. We've got group 1's 20 plants, group 2's 20 plants, group 3's 20 plants. And this group gets no fertilizer, 2 teaspoons, 4 teaspoons. And then at the end, we're comparing the yield, the number of soybeans that are produced for each group. So again, this figure helps with our design. Next, we're going to look at a randomized block design. Now, a randomized block design, it's very similar to the completely randomized design, except in a randomized block design, we're going to have another variable that we are maybe taking into consideration. And that other variable we're going to block together. So grouping together similar are homogeneous experimental units, and then randomly assigning the experimental units within each group to a treatment is called blocking. So like, for example, in my migraine problem, if I were to say group all the males and the females together, those could be blocks. So I have the males in one group, I have the females in another group. And then of the males, I'm going to give my different treatments. Some will be control group, some will be the 100 milligrams, some will be the 200 milligrams, some will be the 400 milligrams. And then I'm going to have females, that's my other block, and I'm going to do the same thing. Some of them will be control group, some of them will be 100, some of them will be 200, some of them will be 400 group. So I'm looking at 2 blocks because it's possible that there could be differences in the treatment for a male than for a female. So that's the idea of blocking. Each group of homogeneous individuals is called a block. So there's some commonality, there's some reason, some other variable that I am blocking by. A randomized block design is used when the experimental units are divided into homogeneous groups called blocks. Within each block, the experimental units are randomly assigned to the treatments. So again, like from the males, some get each of the 4 treatments. Of the females, some get each of the 4 treatments. So I'm looking at the 2 blocks separately. So example, suppose the 60 soybean plants were actually of 2 different varieties, 30 Kimgrow plants and 30 Pioneer plants, and that the 2 varieties have different yields. Design an experiment that could be used to measure the effect of level of fertilizer while taking into account the variety of the soybean. So again, now our 60 soybean plants are of 2 types. We have Kimgrow and we have Pioneer plants. So we can treat those varieties as the blocks. So taking a look here, in a randomized block design, we're going to divide the 60 plants into 2 blocks. We have the 30 Kimgrow plants in block 1, and we have 30 Pioneer plants in block 2. Within each block, we're going to randomly assign 10 plants to each treatment, A, B, or C. So for example, for the Kimgrow plants, 10 of the Kimgrow plants are going to be in the control group getting no fertilizer. 10 of the Kimgrow plants are going to be in treatment B group, which is getting 2 teaspoons of fertilizer every 2 weeks. And 10 of the Kimgrow plants are going to be in treatment C, which is going to be getting 4 teaspoons of fertilizer every 2 weeks. And then the same for the Pioneer. 10 of the Pioneer plants are going to be in the control group receiving no fertilizer. 10 of the Pioneer plants are going to get 2 teaspoons of fertilizer every 2 weeks. And 10 of the Pioneer plants are going to get 4 teaspoons of the fertilizer every 2 weeks. So if this picture here I included from our book because it kind of gives you the visual of blocks, right? So we got 10 Pioneer plants here getting treatment A. We have 10 Pioneer plants here getting treatment B. And we have 10 Pioneer plants here getting treatment C. And then we also have 10 Kimgrow getting treatment A, 10 Kimgrow getting treatment B, and 10 Kimgrow getting treatment C. So that's sort of the idea of blocking. The logic is that each block contains plants of the same variety. So plant variety does not affect the value of the response variable. So we're basically controlling that. The figure below illustrates the design. Notice that we do not compare the yields across plant variety because this was not our goal. We instead know which variety yields more. The reason for blocking is to reduce variability due to plant variety by comparing yields within each variety. So again, we have our 60 plants, 30 Kimgrow, 30 Pioneer. So we have block 1, which is the Kimgrow plants. We have block 2, which is the Pioneer plants. So for block 1, we split those into each of the 3 treatments. 10 get treatment A, 10 get treatment B, 10 get treatment C, and then the same for Pioneer. 10 get treatment A, 10 get treatment B, 10 get treatment C. And then we're going to compare the yields. So for the Kimgrow plants, we're going to compare the yields, and for the Pioneer plants, we're going to compare the yields. Now again, we're not particularly comparing the yields here to each other because we already know there's going to be a difference in the yield based on the variety of the plant. Again, in my example that I used, I looked at like females and males, And I may not compare females to males because we might expect that the results could be different based on gender. Now, we might also actually want to compare the yields of KimGrow to Pioneer. If I was the farmer, I would want to know what's going to give me the greatest yield overall. So if that was the case, we could compare all of them. It's possible that, for example, treatment C is better for Pioneer, but treatment B is better for KimGrow. But which one of those is better? Which produces the most? Well, it's possible that treatment B for KimGrow is a better yield than treatment C for Pioneer. So if I was a farmer, I would probably want to buy KimGrow plants and use the treatment B in the future, as opposed to using treatment C for Pioneer. But again, the purpose of this experiment was not really that. It was for seeing for each variety, what's the maximum amount of fertilizer that optimizes the yield. So our last design type is called a matched pairs design. So basically in a matched pairs design, what we're going to be doing is we're going to be matching up individuals. So for example, if I have, say, like twins, I would put one twin in the control group and one twin in the treatment group. Which twin goes in which group would be randomly assigned. So like if I'm testing my medication, again, so I would maybe come up with 20 pairs of twins, and 20 of the twins would go in the control group, 20 of the twins would go in the treatment group. So what's the purpose of that? Well, if individuals are twins, then we would expect maybe that they're pretty similar, except possibly for the medication. So I'm controlling a little bit more. Now, it doesn't have to be twins, but there's some kind of matching. So sometimes the matching can be as simple as what's true before treatment versus what's true after treatment. So it could be the same individual before and after. So for example, if I wanted to know, does exercise affect cholesterol level? Seems reasonable that it might. So what I might do is measure an individual's cholesterol level and see what it is. So that's before treatment. And then I could put that individual on an exercise regimen for, say, 3 months. And then I test their cholesterol level again at the end of the 3 months. So I have their before exercise cholesterol and I have their after exercise cholesterol level. And so I can pair those two things up to see if there's differences. And if I do that for a large number of individuals, I have their before and after cholesterol levels, then any difference in the before and after levels could possibly be attributed to exercise. So again, it could be that we're matching individuals up, or it could be a before and after kind of scenario. So a matched pairs design is an experimental design in which the experimental units are paired up. The pairs are selected so that they are related in some way. That is, it could be like the same person before and after treatment. So like my cholesterol example, it could be twins. It could be like a husband and wife. It could be people from the same geographical location, and so on. There are only 2 levels of treatment in a matched pairs design. So those levels of treatment are going to be control group and treatment group. So there wouldn't be any 100, 200, 400 kind of scenarios. It would either be medicine or placebo. That's it. Treatment group or control group. So, for example, an educational psychologist wants to determine whether listening to music has an effect on a student's ability to learn. Design an experiment to help the psychologist answer the question. So we're going to use a matched pairs design. So we're going to match students according to IQ and gender. So for example, we're going to match 2 female students with IQs in the 110 to the 115 range. So now I'm only giving you one example there, but so I have 2 females with similar IQs. I'm matching them up. Then I would have, say, males with similar IQs, and I'm going to match them up. So now I'm going to have more than 2 individuals or 2 pairs. I'd have maybe like 100 pairs of individuals, but I'm matching them up by IQ and gender. So for each pair of students, we're going to randomly determine which students assigned the treatment and which students assigned the control group. So Which students assigned the treatment of a quiet room or a room with music playing in the background? So the only difference, again, we have the same gender, we have the same basic IQ. So one of those individuals is going to go into a quiet room. The other individual is going to have music playing in the background. Each student will be given a statistics textbook and asked to study a topic. After 2 hours, the students will take a short exam on the topic. Compute the difference in the scores of each matched pair. Any differences in scores will be attributed to the treatment of music. So again, say the person in the quiet room scores 5 points less than the person in the music. And then for the next pair, the person in the quiet room scores 4 points less than the person with the music, and so on and so on and so on. I look at that for each pair and see if there's any differences. Any differences would be attributed to the treatment. It's possible that the people in the quiet room could do better on the test. It's possible that people in the music room would do better. Or it's possible there would be no difference at all. So we do have a figure here that's illustrating the design here with matched pairs. It's fairly straightforward. We match students according to gender and IQ. We randomly assign a student from each pair to a treatment. We administer the treatment and exam to each matched pairs. And for each matched pair, we compute the difference in the scores on the exams. And then ultimately we would perform inference to see if we think there's any difference.